I'm Josie, this is Ingrid, and that's Jody. So today we're going to talk about biotoxicity. And so what's biotoxicity? It's basically the biotoxic nature of particles. And so this week, what we did for one of our labs was to use silver particles to try to kill yeast cells. And this is so basically this is really useful in our actual lives because we can use nanoparticles to kill microorganisms, and it can actually save lives.
Okay, so I'm gonna explain this lab procedure a little bit. So basically in this lab, we put yeast cells in different Erlenmeyer flasks and we monitor the number of yeast cells by using a manometer. It's basically a tube and you fill it with a little bit of water. So when the yeast cells do their respiration after they consume the sugar that we put in, they would like either produce some carbon dioxide or they will respirate and then consume oxygen. So the water level will rise or decrease. So that's how we measure. Yeah, and so we basically put 3 kinds of silver into the flasks, which are silver nanoparticles, silver powder, and silver ions. And so silver is toxic, so as it gets to the cell, it ruins the cell membrane, eventually kills the cell. So I'm going to explain a little bit about all 3 kinds of silver. So we have silver nanoparticles. Nanoparticles are usually 1 to 100 nanometers. So in this case, the silver nanoparticles are the most effective because they enter the cells and kill them by releasing silver ions. And we make silver nanoparticles by mixing 2 solutions, silver nitrate and sodium citrate. And then we'll heat the solution until it turns a faint yellow color. And then silver powder is just silver powder. It's powdered silver, and it has a silver color. And then it is the least effective because the particles are really big, so they cannot enter the cells to kill the yeast cells. And then lastly, we have silver ions, which are silver atoms with a charge. So these ions, because they have a charge, they have to go through special ion channels. So they take a long time. They're not as effective as silver nanoparticles. So we're basically measuring how effective each silver particle can kill the cell. So let's get onto the picture. So here we can clearly see that we have 3 different doors at the airport. And then these people represent different kinds of silver particles. So this guy right here, he is big and he is carrying a bunch of small human with guns apparently. And try not to bring guns with you to the airport, you'll get in trouble. So so this guy basically represents the silver nanoparticles because the silver nanoparticles will carry the silver ions, which will kill the cell. So they're trying to kill people in the airport. So, and also notice how he is wearing a yellow jacket, which is the color of the nanoparticle solution. And then move on to this one. These people are just like these tiny people. So they are just silver ions, and they're also carrying the same goal, which is to kill people. And notice how here the artist points out that they have transparent shoes, which is the color of the silver nitrate solution. And then here, this big guy right here, he is obviously too big to enter through this door. He also carries a gun right there. So this guy represents the silver bulk particle, the silver powder. And notice how he's wearing silver shoes. So it is the color of the silver powder. So so at this airport, everyone is trying to kill people. So so that is what the silver ions are trying to do in the experiment because they're trying to kill yeast cells. Yeast cells are the people. And then notice how the silver nanoparticles, they can kill the most people because they're the most effective. And then here, this guy kills the least people because he is too big to even enter the airport. And I'll carry it on to Jody.
So this picture is really good. But one thing that we did in the lab that we saw is that how fast and effective each size of silver particles are. Um, the silver nanoparticles on the left side is the most effective and the fastest to kill the cells, while the two on the two on the right are the slowest. So you might be wondering, why are silver nanoparticles so much effective than the other two types sizes? It's not the smallest, but it's not also the biggest. So what is special about it? Well, the main reason of this is because silver nanoparticles go through endocytosis, which means basically the yeast cell sees it as food, and then it disguises itself as food, and it enters the cell directly and killing it. And this process occurs much faster than just ions going through a different channel and entering the cell a different way. So that's why it's really effective. The other thing is high surface area of silver nanoparticles, because silver ions jump on the cell a lot, and it constantly dissociates and reassociates. So with more surface area, the silver ions are able to be more active and dissociate more, and therefore releasing more silver ions and killing the cell quicker.
Somebody else has to ask the question first.
Hello. Okay. So basically, in hospitals, they use this silver coating to coat their equipment, and it actually it releases small amounts of silver ions, and it helps to. It's basically an antimicrobial coating for it, and yeah, it helps kill cells and.
Any other places that you get silver nanoparticles? Can get them?
Cancer research.
You can buy Band-Aids. You can buy gym socks.
Yeah,
you can buy keyboards. Anything that might get really gross and stinky or get touched by a million people, you can now buy with silver nanoparticles embedded in it. Okay, so I get my question. So you comment that the nanoparticles are wearing a light yellow jacket and that you made your particles yellow. So why are they yellow?
I think it's because of how they absorb the light. Yeah. And so there's something called oscillating frequency. And then when electrons on the particles shift on their electron clouds at a frequency that is the same as the wavelength of the light, it absorbs it and then it gets a color. So it's yellow in this case.
That's good. All nanometals are colored because the electrons can just slosh back and forth and absorb light. So, good job.